Ready to roll. All right, thank you. Okay, well, I'll do this quick. You know me, I'm Dr. Lisle. Thank you for coming for these both in person and those that are on the Zoom. I want to talk just a little bit about applications in healthcare for computer science and AI. This is the overall agenda. I'm going to present 3 different use cases. One in radiology, which is a lung cancer research work I did with Rutgers Hospital. Then in pathology, some AI segmentation and some downstream models that we talked about there. And then a microscopy application, computer vision and AI techniques, used to really help the scientists understand their data better. And along the way, we'll talk about AI trends. I'll try to get rid of this little screen sharing thing. Put it down here on the bottom. It's lovely to see it that way, right? Okay. Maybe we'll find out where it's going to be the most out of the way. So the Lung Cancer Research Project, this was funded by Florida's James Nestor King Foundation. Work was done along with UCF at that time. And the goal was that we had radiation oncologists in Orlando Health that had by hand processed their data. And they had hand segmented these regions and during their initial test before they actually treated for radiation. But if ever about 3% of the cases when they treat radiation, they miss, and the cancer regrows. And so the point was, could we give you our time sequence data and tell us which ones recurred? Can you train AI to determine more quickly when the cancer recurred? Because they said by the time I could see it visually in the next CT scan, I could have treated a couple months before that. I would have likely found out. So that's what they asked us for. We developed an image-based metric that helps for survival analysis. Some of that long-term goal, we're still waiting for them to finish annotations, but we'll discuss that. All right. Okay, are we going? Okay, there we go. All right, so this is the cohort description. We were given 230 patients, and each patient had had the original, lung cancer lesion and the organs that were hand delineated by the radiation oncologist. They had done the structure boundaries and that kind of stuff with it. This is a longitudinal way to look at the cohort. They were detected using PET scans that this was some cancer. The orange lines are when we did a CT, when we know we're going to treat for radiation, so we do a nice quality CT scan to use that to figure out where all the organs are, figure out where our lesion is, decide what to do our target planning is. And then follows every 6 months or so. And that's something that's exciting. Now, at the time, as I told you, that surgeon hand delineated all the organ systems and where they were going to target. There was no time to do that for the follow-on scans. So they didn't have any way to automate anything else. They just looked at the raw CT scans from then on. But AI has come in the meantime, right? For things that are like, anatomical regions, we understand that we have a lot of data for that. Open source models like the Total Segmentator are very good at it. So I took the cohort, 167 patients out of the cohort, just automatically ran this segmentation system and categorized the overall volumes of the different scans, the labels that it discovered. But lung cancer lesions are one of a kind, right? So there was no way to have a pre-trained model that would know necessarily how to do those. So we trained our own for the lung regions using the original data from the radiation oncologist. Trained a unit R model that came from the Menai framework, which is popular in medical imaging. I'll talk about that a little bit more in a second. After training, we just had good accuracy with this. Not the world's best model, but it was good enough for what we needed. It was easy to deploy. Menai is an open source project that's focused on medical applications. It's supported somewhat by NVIDIA and a lot of I think King's College, Cambridge, and they're trying to find, they're picking and choosing from the best current papers. Like, okay, this is the unit R model, we like this one, we'll take it and put it in our framework. So they're just finding the best models that they can get and kind of build it into a framework with the idea of medical opportunities that we both interesting. Yeah.
The algorithm is after your first discovery of cancer subsequently, or you have some prior data prior to when cancer is?
In this case, it's afterwards. And we were given the scans for the initial detection before treatment. Yeah, that's the green side. I haven't looked at those yet. I've been looking at the scans and the progress afterwards, because that was what I was asked by the surgeons to say, we want this tool to evaluate every time, just automatically look at our progress every 6 months and see what it would be. That's what they asked us to do. There are certainly, these lesions could certainly have been more quickly discovered using screening. And the data in that, there was a national screening trial that definitely is beneficial to do. So there's reasons to screen at a time. So, the standard unit model is on the left. This is just popular in medical imaging for anybody. It's a popular AI algorithm. It's not the very best for most, but it's easiest to put in your image in the front side, have it generate latent features, and then down the sample, you have latent features at different levels of resolution that merge together, and you can produce a good quality segmentation. It's not very expensive to train. So in the medical community that I'm associated with, that unit is a very popular one. I use a slightly more advanced one. The unit R has transformers in the front instead of convolution. And the problem with CNNs is there's there's a restricted range of influence. So if there's very disparate parts of the image that are related to each other, the CNNs can't see that. There's a region limitation depending on the size of the convolutions that you use here. You're only sliding your convolution window around neighboring pixels, whereas the transformers can keep track of things in distant parts of the image. So it's just a better model. Same idea, you're downsampling and bringing latent features over different resolution. So this is the way that training worked. For anybody that's not that familiar with training, you're using a loss function, you're minimizing the loss function, which in this case was DICE, which is the overlap, intersection over union overlap for segmentation regions. And you tested on a validation data set, not the same ones. So we took 20% of our data, put it over to the side for testing. And we got overall 76% chance. This is good for lung cancer objects that are very different and individual. If you had something that's always the same, like a lung, you get 90 or 95% accuracy of overlap. But with these one-of-a-kind things, that was considered good overlap. At the time, I was also working with a doctoral student. What we did was enhancing the way UNet worked by changing the dilated convolutions. This tries to get at that problem CNN I just told you about, the limited resolution, limited region. And then cyclic burning waves are just a better way to optimize. So we could get a couple extra percentage points of accuracy. Then we moved on to self-supervised learning because medical imaging, there's relatively small amounts of data. Well, there's largely, but it's all in hospitals and they can't share by law. And so medical data is hard to get a hold of. So pre-training on a lot of pre-CT scans from different regions is beneficial. So we found pre-training obviously worked within the medical domain. And uncertainty was important to our radiation oncologist because you never know exactly what the boundaries of organs are, particularly here. So we wanted the model itself to kind of be able to show us the regions of uncertainty that we might be doing. And uncertainty is done in models because using what's dropout layers. During training, dropout layers, most of you are probably familiar with it, are used to make a model more general. And if you think about it, like if you're on an assembly line and the guy next to you always knows how to do that widget, and he's always there, he's always doing it, then you don't really learn that very much. But if some days he's not there, you kind of have to cross-train and know how to do his job as well as yours. Same with the neurons. If they're all of a sudden not there during training, then there's a generalization that happens by the neighboring ones. That's during training, it makes for better neural network models. However, if you turn on dropout during inferencing time, when training is done, then you induce the stochastic variance in the output of the model. And that's how you get your uncertainty. You run the model 10 times with different neurons turned off during the runtime, during the forward inferencing stage. And then you average your answer and you get your uncertainty that way. So that's how you get medical professionals to buy into the AI systems better because you can produce the kind of uncertainty that they're used to seeing and need to see. We had a temporal data set. So this is, starting in 3, 6 months, 12 months afterwards. You can see that they were able to reduce the lesion. But each of these, after the first scan, this person went to Disney World and went to Michigan and came back 6 months later. So these are not registered with each other anymore. So it's hard to actually directly compare them geometrically. So we developed a registration pipeline that segments the lungs, uses the lungs to make a bounding box, orients the bounding box, clips them both, and then does registration. Once you've simplified the problem, you've made sure that actually everybody has the same, because it's the same person or the same lungs, so you can use this. It's not perfect, but it worked well enough to give, sort of very close approximation. There's the original planning CT and 6 months later, and it's pretty close to this alignment. Of the candidates we had, there's still about 90% DICE score, 90% overlap between the tumor regions between the originals and the follow-ups. So it was good enough for us to be able to take those and use those as measurements for the thing. So we kept track of, in our cohort, how many patients had at least 2 or 3 or 4 what I call high quality registered follow-up scans. So we could compare geometrically what they were. And we took those, put them into a web-based annotation system and let the clinicians go at it. Because we wanted to be explainable at this point and say, okay, to the clinicians, okay, how do you determine if cancer is recurring? And they said, we look at things like, is there a bulging margin around the area where there used to be? Is there a pleural effusion, which is like fluid in the bottom of the lungs? Is there opacity increases? So they gave us a list of things that they would want to see. And so we put those in this web system so they could step through their initial follow-on, quickly step back and forth. Okay, I see it in the second time, I don't see it here, I see it, okay, here's where it is, set bulging margin on the third follow-up. So they could give us this temporal data set that we can use to predict with. They're still working this annotation, so this is something that's not finished yet, but this is something they were doing. I'll skip this radiomics. So previous to foundation models, which I'll get to in a second, radiomics was the best thing we had, right? Is you took an image and you just generated 100 different mathematical values out of the image, whether it's gray level occurrence matrices, whether it's suddenly contrast, you just generated a bunch of mathematical measurements, extra features on the image. You didn't know exactly what was useful. But we used that to take, while the clinicians were still doing the annotation, take the radiomics, unsupervised cluster the patients into groups by the radiomics values of their pre-planning CT. The idea is, can we predict from here the survival of the patient over a couple of years just on their radiomics values? And we did that. We took we clustered them, and then the highest value cluster, the cluster that had the longest survival, we marked as, I'm in the member of the longest survival cluster of this subgroup of our cohort. And that feature we used to try to stratify the patients, decided. And in terms of asking the radiation oncologist, and they explained, okay, we have 2 different tests that we use to determine if this is a valid metric that you've created here. And the first one is a Kaplan-Meier survival analysis, and it'll tell us if it's a subpopulation that you've identified is different than the general population. And when we did that, the Kaplan-Meier was successful. And so we were able to predict if you're in our high survival group, according to our radiomics metrics, you did do survive longer. At any point in time, there was a bigger percentage of those in our high survival group that did indeed survive. So they were, okay, that's great. You've got the first test done. We know that you can predict this, but now, is it relevant? Could we have predicted this clinically without you? And that's the Cox metric, is let's look at all the other things we know about these patients. And if we put in an automatic, the Cox metric will look at all the factors we have and it'll weight them. And it'll say, okay, only if it picks yours as important is yours necessary. Otherwise, we could have figured that out by the number of pack years that that person smoked, their otherwise lung function, And it turned out that our metric was more important than everything except for had they already just been treated with chemo, which is kind of not a real measurement in that sense. So obviously that makes more sense in terms of their survival, but everything else was more important than that. So the clinicians said, congratulations, you've helped us figure out from just a CT scan before we treat whether there's a likelihood for 3-year survival or not. Okay, so we took that, and then we also, it's known that if you have trained the DNN model for segmentation, we all know this, that there's low-level features up to high-level features that a multi-layer perceptron or a model learns. So if we were to go and pull features from near the end of this classifier out and add them to our radiomics, it should in fact make a better result. I mean, we found that that's the case, that we were able to produce a couple percent extra selectivity by pulling features that were trained on the segmentation. So we took our segmentation machine and just pulled that from that.
So what is radiomics?
It's the radiomics, before we had foundation models, just like fractals based on the image. What's the longest?
So different features.
Yeah, just random mathematical features. Yeah. And so that was all we had for features beforehand. So it was the rage 5 years ago to about 2 years ago, or whatever. So we were on that rage at that time, and succeeded using it. but it's obviously better to have foundation models now. And so I took that project ended, but I took, just as an example, I'm using what's called the MHub project that's out of Harvard. That's a standard way to run mostly medical models. And it packages them, put them in containers, make them easier to use. The idea is, a lot of times you do a great thing, write a paper, get your paper published, and then the code goes away and no one else can use it anymore. This is the idea to make them all reusable. So Harvard just released a foundation model for lesion detection, for lesion identification, like 12,000 CT scans. So I took that, clipped our boundaries, and just used it to generate 4,000 features, which should be more interesting, more latent information, much more information dense than the random radiomics. And within a couple hours, I was just able to put a logistic regression model on it and get a good-looking result model for the survival. So this survival model, with a couple hours of work, was better than our week's worth of work before with radiomics and pulling things out of the model. So that this is one of my, I'm preaching to the choir. Foundation models are very exciting in terms of how they can help us develop task-specific models easier now, and more reliable models, the more generalizable models. I did try plotting the latent space of that foundation model, and in the course of the couple hours I had, I didn't really see any localized, 3-year survival and under 3-year survival. I didn't do any fine-tuning at all. I'm sure if I had fine-tuned this model with these metrics, with these images, we would have seen some organization here. But I wanted to just show this is the idea in terms of what you might see and what you want to look for. So,
Can I just add a question? Just at the prior slide.
this one?
The original Pulse Foundation model was 11,670 CT scans. Those are for all diseases.
For not all, but for not just lung cancer, yes. They had a set of about 10 different types of sarcoma and some cancer devices in different organ systems. And they actually just pick the area right around it. So it's not the whole thoracic unit. They just pick an area, 200 pixels on a volume around the lesion and use that. So this was not, it was not just ours. It was a mixture of ones. So, okay. And so our idea was, given that we have planning CT, given that we know what the radiation dosage is going to be, and that we know some clinical outcomes we can train, and then when it's time to have a new patient, they come in, they get their scan, the radiation oncologist says, okay, this is the plan I want to have. And then we can ask the model, given these inputs, what are you going to see? Are you going to get to, sorry, one more time? I did it wrong. Okay, there you go. We'll get, given a patient-specific feature, because given this particular geographical or geospatial relationship of the radiation dosage, the geospatial relationship of the original imagery, we'll get good recovery, or we'll get bulging margin at 6 months, because we use those annotations to train a time sequence model to predict it. So this is our outcome, or desired outcome. We've resubmitted for additional funding to finish doing this annotation work.
So this traditional, the radiation plan, what is that? Another image, or it is the text, or what is it?
We do have a 3D image of radiation intensity at every voxel in that 3D space that's registered to the original planning CT scan. So we can make that an extra channel to the original image.
And these clinical outcomes, and what are these?
These are textual data instead. So this is just tabular data that we do here.
So what is this deep learning AI? What is it? Will take both text and images or something? Which one is that?
Yes, the easiest plan. So this is a hypothetical one this way. What you're talking is, yes, what we would want to be doing is late binding. We'd do the imagery, do that prediction, and then do late fusion and do the prediction afterward. It's a multiple step. Yes. And I submitted, we didn't get awarded to ARPA-H a year ago. They asked for a system to allow you to trade off between late binding and early fusion and late fusion is what they called it. Here is early fusion, where we'd have to convert these to be the same domain, and so I didn't win that one, but maybe next time. So, places that we can extend this research, there's obviously better foundation models, the segment model, there's a med version of the segment model right now. If I did that foundation model with MedSam, it would be stronger, I'm sure. This is what I just discussed, throwing the planning CT and the dose volumes together to do predictions. Why not look at temporal changes in the latent space of the foundation model is producing? Because it's producing a very good embedding of that three-dimensional view of the patient. Right? And so there's a lot of dense information in that 3D view of the patient. And so why not look at those changes temporally in that 3D view? There was, when radiomics was the only tool we had, then there was delta radiomics, which is, okay, have those changed. But the latent features of the foundation model are so much more information rich. So, okay. Something else I would like to do in the radiation, radiology space is making things more intuitive to the end clinician. Why not take the embedding information from the imagery and mix it with your clinical data, put that into like a KnowledgeVis. I did this as a little prototype. I took the clinical data, put it into made a little graph out of it, put it into a RangoDB, and used LangChain on top of it and asked ChatGPT. And so I asked it, okay, how many patients am I have this tumor size? And the language model converted my query into graph query language, ran the graph, and then took the output back and put it back into text for me. So it could tell me which patients were the ones that had the right outcome. So if we had the really embedding here, richer information in this graph, if this was a knowledge graph, then I'm going to suggest that this is kind of the new RAG, right? We used to just take embeddings for lots of documents, and ChatGPT does this, right? Where it has a database of embedded for similar documents on the side. Well, doing instead of RAG, doing a knowledge representation that you can use would be very powerful. So I think this is something that could be. And if you mix this with a federated learning kind of thing, and you have sets within each hospital, they can all understand their own data, and they could exchange some cohort level data or model level data that's safe to communicate. So, all right, I'll skip that for one definition time. Let me talk about digital pathology for a little bit. Radiology is relatively mature. All the systems read and write the same information at .com. They all know how to coordinate with each other. Radiology is still coming along. And I was surprised when I talked to Orlando Health's chief pathologist who said, nobody in Florida is using digital pathology yet. We all just look at our microscopes and we store our slides in our slide archive. And he said, my friend is the chief pathologist at UMA, and they're just using physical slides too. And I'm like, really? But digital pathology is on its way, and there is now a standard that connects with .com so that you can connect a patient's radiology work along with their pathology work. The problem with this, these images are giant. 150,000 by 100,000 pixels is a normal size image. It's 2 gigabytes to 5 gigabytes to more on an image more on a disk for a single image. So they're usually represented as pyramidal. So if you want to look at the whole image, you're just looking at a downsampled version all in one file. But models usually have to pick a particular resolution. So we'll pick one resolution out of that pyramid, like 10x or whatever, and use that to train with. Okay, so I worked on a team that thought of this rare cancer called rhabdomyosarcoma. This is a rare pediatric cancer, and it has 2 subtypes. We call them AMRS and EMRS. The alveolar, the AMRS, is the more serious of the 2 subtypes generally. But there's a case where you can have the less serious one and a particular genetic mutation, and unfortunately your prognosis is really terrible. So it makes sense this is a specialty disease, so it makes sense to have some tools that pathologists who are not specialists on this could have access to these tools. Because if someone comes in and happens to have this particular genetic mutation, then treatment as quick as possible is really, really intensely needed. There's terrible results if you don't identify this quickly. So we wanted to find ways to identify the subtypes and ways to identify this genetic mutation. This is a group, there's a group at NCI that's leading this from a biology standpoint, and myself and a couple colleagues are doing the AI part of it. So this is how we did the AI flow. We take H&E slides is the most common staining for pathologists. There's lots of different kinds of stains for different antibodies that are being expressed and all that. We can talk, Dr. Zhang and I talked about that for a half hour this morning. but H&E is the most common one. And so our research question is, if we could segment a tissue from just this H&E biopsy, this like a biopsy, and if we could determine the cancer subtype and their survival and everything else on their genome, all on this H&E slide, that would be a perfect tool to be able to send to remote locations and remote hospitals. So we designed a process. We would segment first, get out these 4 classes: AMRS, EMRS, stroma, which is connective tissue, and necrosis, which is tissue that's dying, right? And then we use the output of the segmentation to do some other basic models. And for interest of time, I'm just going to talk about these 2 real quick. The segmentation and this particular genetic mutation that's bad if it happens. So here's how we did the segmentation model training. It uses a regular unit, not the unit R that I had done before. It's a simpler unit. We use an efficient net encoder on one side. That wasn't medical image trained, it was just ImageNet trained. So that was just lots of general imagery. But remember, in a multi-layer neural network, the first layers are simple, low-level features, so it's okay. We use thresholding because here's a giant slide on that last picture, right? Of this slide. This is background tissue right here, right? You have to work your way across this slide because the slide is so big that the model's just taking tiles at a time. Well, if your tile is mostly background, then it's a waste to send it to the model. You don't want to train it because you have one label for the slide. This is cancer, or this is MYOD positive, and you don't get much information about cancer in this background slide. So you have to figure out how to do the multiple instance learning problem in AI, which is to take samples. So we thresholded here to large amounts of tissue relative to background before we sampled it. We took matching patches, put it into a unit. This produces a 5-tuple class probability for every voxel. Is the mixture between AMRS, EMRS, stroma, necrosis, or background. So this model produces this high probability, and then you threshold it depending which is the highest probability if you want to have a binary segmentation image of this is what it looks like. This is what a sample is here's the original image on the left side right over here. And I picked this one because it's an image that messes up the model, so I'll show you why in a second. And our workflow was that the expert pathology gave us this annotation of here is stroma and here's cancer. Okay, now there's a couple things missing here. This is not particularly accurate compared to this tissue, because if we just sent the model this background area and his cancer diagnosis, then we're confusing the model, right? And we know by now from our training that large amounts of data, even if it's somewhat noisy, is better than small data, but clean data is better than noisy data in general, right? So we, in part of our segment workflow, we did an exact intersection so that there's no cancer anywhere that's not, so anywhere with a tissue, an exact tissue intersection. So we reprocess these to make them, background where there was background and everything. So the model produces this 5-tuple here with sort of uncertainty at the boundaries. You can see where it's dark here. We pseudo-colored this to show you the classes. But you can see there's kind of the probability of changes between them. And it gets confused because here is the problem with pathology. This tissue is folded on itself right there. So it's bad data that we're passing it originally. So the model gets a little confused. But when you look at all these cases and how we did across the 304 cases, it's really quite strong. And the pathologists have looked at this. We like the quality of this particular model. So you take the output of this probability image for the segment, and then we used it along with the original image as input into our genetic mutation model and also the survival risk model. The architecture of this one is very similar to that one, so I'm just going to present one of them. And so how we trained the MYOD model, that was a ResNet 50, just a classifier, because we're trying to get out just a relative risk of genetic mutation given this image. Right? So this bad mutation occurs in only about 3% of the cases. So we have a data imbalancing problem. So we over-augmented on the MYD1 positive patches. We took 4,000 patches from every input. There was only 9 slides of positive cancer that we had out of the hundreds we had gotten. So we added that, so we took 5 times as many patches from those cancer MYD1 positive as the regular wild types. So we can present relatively balanced information to the model. And of the 9 positive cases, the model predicted all positive cases. So it was completely accurate in true positives. It produced a couple false positives also. which is better than false negatives if you're doing a medical pre-screening. So, and it identified 42 of the 45 negative cases correctly. So this could be a tool that could help pathologists by saving a fair amount of time. And here's the for sure, here's the marginal, please check these also. Here's the ones we're really quite sure are not positive cases, so prioritize these first. So, for the small amount of data that we have, we think this was useful. There's also some, there's issues here. This is, I was going to add one more slide that talked about the real world data that's messy because of our positive MyoD1s. This model produced different levels of risk for all those. So we have to decide where do we draw the line and say it's positive. But our clinicians that read this work agreed that we did it right. During inferencing of the model, here's what we did. We took the segmentation, we sampled 4,000 patches out of the output of the segmentation model until we had 4,000 patches that were positive for this cancer and passed all those through our trained MyoD1, our trained ResNet, and averaged that risk value out to come up with a risk value for the overall slide. So the random sampling in there to try to address the multiple instance learning issue and address the fact that there was enough cancer. A little bit wouldn't be enough to train this. We took the average and thresholded our average.
So the reason we're not able to use the whole image because it's too large or something? Why do you use the...
It's both large, but also that's these remote parts of the image where we identified there was no cancer. We also think is not valid to send to this model that we focused the training goals of this model on just dense patches of either MyoD1 positive or wild type. So we didn't want to pass a lot of wild type patches into the system.
But you don't know, right? You don't know which page has a cancer, which is not.
Correct.
So randomly pick, and you may end up with most of them non-cancer.
Yes, you could. And that's why I didn't try this as a separate thresholding afterwards. We get the overall average, and then we thresholded that to decide if we saw enough positive ones, then we passed that threshold. I also set this up with a little website and whatever to be done, so you could upload your image, and I would produce that with a little graph. That was the one page that I didn't have time to show you. I didn't have time to put into this slide. I think
what will happen is, for example, you send the whole image. Can you do that? Can you?
Yes.
What's the resolution of this image?
150,000 by 100,000 pixels.
So that's,
you could send. We'd have to recalculate the thresholds.
Yeah, probably. These are starlit images. Right?
Yes, correct.
Yeah, those are known to be very large, right? So that's the reason. That's what.
Yes, that's why we're sampling it. We could do the other ones. We would just have to recalculate all the thresholds because it would be. And we figured that we would be better at the signal to noise ratio if we sampled only within the cancer regions. Yeah, good question. So now that foundation models are around, we're redoing we're redoing the subtype model. The subtype model was a ResNet 50 that we trained before out of tissue microarrays, the little TE circles. I'm currently right now taking the Virchow1 foundation model, which is from Page AI. It's one of the better pathology models that's out there. This is open source on Hugging Face if you want to go get it. And using it, I processed those RMS images and generated embeddings, and they give you 2560 feature vectors, which is much more information dense than the radiomics ones we had before. Right? So this could be just used as the features to train a subtype classification model. So this is in process right now to evaluate how good it was compared to a natively trained from the image model. And my assumption is that we're going to see this current ability to replace, specifically trained models with foundation models and classifiers. Okay, just a couple slides real quick on neighborhood analysis. A different project that I relatively started in working. This is an HIV model where this is an actual image that's been annotated by our biology colleagues. And the danger with HIV, usually when we get a flu bug, our immune system fights it off within a couple of weeks and we feel fine again. Unfortunately, HIV knows how to hitchhike on top of our immune system and turn some of our immune system cells into virus producers. And so that's what happens. This is a mucosal layer, there's a boundary. Viruses try to get in. Maybe where it was weak right here, they did manage to get in because these yellow ones are RNA-seq. They're immune cells that have been converted by the HIV into virus factories. The request from us, from the AI, was from this biologist, okay, can you help me understand the networking that's going on here? What's happening at this conversation? Is it because these were all thin here? What percentages of immune cells are converted in a neighborhood? Can I figure out how to predict that? Can I figure out if there's relationships here? So they give us a small number of slides, but they're very heavily annotated, where they've looked at the measurements here and stuff. So this is exciting. It's a whole different aspect of understanding how to apply AI tools. And I'm bringing this up because here's a different way to apply AI tools. And we're used to taking a problem and training a model and looking at the model and producing good papers, and then all that's necessary. But if you have this wild biology problem, this is more background than a graduate student has time to understand and learn and practice. So I've used this open source tool called the multiple choice microscopy environment that's out of Harvard. And it uses embedded models like this. It's called unmixed right here is the way they call it. That's an image segmentation model that takes the boundaries of images and boundaries of cells and decides how to make them into actual cell segmentations. But what they found was tissue is not always, sometimes it's thicker than other times, so there's blurry areas instead of fine areas sometimes in these. And so this has been trained with original images and images that were blurry and images that were not blurry. And they found that actually that ends up with the resulting better. And that makes sense. The population using to train is better than the population we're using to test with, right? So you can plug in, and all these interfaces are published. So you can bring in your own segmentation models and still use this machine to just push a button and work through the workflow, which is handy because then this is a production system that we can insert. We can take our computer science expertise and plug in and still let the production system do the production work. And so within an hour of taking an ROI and running that system, I was able to take a look at it and ask it to qualify the type of cell types for us. And so of the cell types that it discovered, it classified them as 9 different types of cells and gave me the counts of those different types of cells that it was segmenting. And in microscopy, in this type of world, there's a lot of different stains. So this is a 20-channel image rather than a 3-channel image, right? And these can be 100 now, whatever the newest machinery. And to find a particular type of cell, it's a and or. If this is true and this is true and this is never true, then it's type CD4. So there's an algebra that they use to figure out which cell is actually which. Because each of these channels is generated by staining a different stain that reacts to a certain chemical. So this is something that, and when we sent this information back to the biologist, she's like, I've never been able to see my data this way before. Thank you. They had by hand annotated. They were able to see this. So we were able to show her just, here's where your CD4 cells are. Here's where and this is the CD4 is the target for HIV. So this is one of the ones that gets converted and becomes a factory. So we were able to plot the neighborhoods of those and the conferences of those and the locations of those with them. So I like that. I also think there's a place in this particular domain to use graph neural networks because the locations of the cells and the cell types and the distances, this is a problem that matches well to, I would say, a network data structure. And so using graph networks would help us be able to really extract more interesting information. And then I could see the idea of being able to detect neighborhoods using the latent space results of the networks better. So this is an area that I thought was really interesting. Another project we had, and Dr. Zhang and I were talking about this morning. Over on the far left, the microscope gives you a pretty blurry image. So there's confocal microscopy, which is very precise, very pretty, very slow, and very expensive. And so a lot of scientists want to go to what's wide field. This is 100 times more faster, 100 times cheaper, but the imagery is not quite as good. And so their question to us was, okay, can you make our wild field image as good as it would have been if I had done a confocal microscopy image? And the answer is we did. We generally used a CycleGAN and did it basically 8x upsizing. And the original image, when it's run through CellPose, which is the best AI model for segmentation right now, you can see that it misses a fair amount of stuff. When we took the same image and super resolutioned it, then the CellPose algorithm was much more able to catch things that were here. So we have a bioRxiv paper that's about to come out that talks about this so this is now being put into production within our group of NCI, because he's like, this gives me the ability to now use these inexpensive, fast microscopes to do my problem instead of doing confocal. So this is something that I think is a very useful application of the CycleGAN. It took us a little while to train this GAN. We took confocal imagery, sampled it so we had high and low and accurate, fuzzy and local, and trained the CycleGAN to be able to get good product. And
so the CycleGAN is known method. So did you guys just took that and use it here, or you did something on the top of it?
We actually just employed it as it was. This is the standard CycleGAN. There was a lot of creativity in terms of how we had to present this data to be able to get the results we wanted. But we turned out to not have to innovate over the CycleGAN. And this is not even the best GAN, as you know. I started using a couple other ones. We found the more recent ones, I'm trying to remember the name of the one I used, worked well in 2x and 4x sizes, but after that we started to get artifacts when we tried to go. And he really asked us to please try to get to 8. Please try to get to 8. So we were able to. But so that was just a use for this off-the-shelf CycleGAN. Okay, quick answer for takeaways. I've said this a couple times already. I think foundation models will really ease the use of the need for large annotated data in the future, right? I think that they'll be able to help us make better problem-specific models with less engineering time necessary. I think AI models are already ready to help clinicians now in finding cancer. Our models for lung cancer, I told you, but those were beneficial already. There's still, academic and political reasons that they're coming in slow, but demonstrating them and having partnerships like I think UCF is already doing with the local schools and hospitals could allow us to take these research types and put them in places where they're really helping people originally. I think that the large language models, they're already ready to take clinical dictations. A couple companies that are doing dictation and stuff, but coupled with the knowledge graph, like I showed you a couple slides back, I think is very interesting for the evolution of RAG. Without the LLM, the engineer would have to have created all these queries by hand over their knowledge. And clinicians are like senior faculty; they're too busy to do detailed things like that. So let's give them tools to do that. I think graph neural networks are interesting. So I really just think that the AI technology is accelerating. It's continuing to explode. And transformers are being replaced with Mamba, some other, so we're going to continue to have much, much more powerful AI systems. So let's continue to make those available, and with industrial partnerships and med school partnerships and others, let's get them in the hands of people so that science itself can be advanced more often. I was talking to Shoshana about the collaboration with physics and about using the molecular confirmation, AI in predicting molecular conformations. That's exciting because I did a lot of parallel processing for molecular docking years ago. So, for anybody who's not, you all are AI familiar, but if there's students that you have that are beginning or whatever else, there's a couple easy ways to do it with no-code environments to get used to that, and also just those are handy. So those are things. There's some recent publications and there's some funding history. So thank you. I had to race through those. Sorry we got a slow start, but do you have any questions in particular about that? Either first from our local audience, and then in Zoom I'll ask you guys in just a second if you want to ask too. Anybody in our local audience have questions? I think you did along the way, but feel free. Yes, sir.
So in the local campus part, we're talking about building a visual vocabulary. Did you have a plan in place to build that visual vocabulary?
Yes, the clinicians didn't get enough time to finish, but we did, that picture I gave you of the web system, it did indeed run. It's up, we loaded the whole data set into it, and they came and started to annotate, and they said, this does work for us. We could finish doing that. And they said, these are the things that we look for. And it took me a while to explain, sure, it took me a while to explain to them that, okay, just because you can see it in 12 months afterwards, tell the model that, but now I can go back and I can back up time and I can run from my training. So I can tell you, this case will develop that in 12 months. He's like, well, I can already see it. Why would I need AI to help me? I'm like, no. you can project ahead of time which path this patient or this group of patients is on, if you finish this annotation for us. So that, I found that to be a surprising, hard to explain. But once we explained it a couple times, they were like, okay, we do understand now. And we see the reasons for us to spend some time finishing that annotation for you. So, yeah, good question. Anything else from my in-person company? Okay, quick. Any questions from the group that was able to join from Zoom?
I'll just make a comment. There's a gentleman named Andrew Barkowski at Tampa VA who's really emerging as one of the national leaders in VA AI, and he's a radiologist and has talked about this a lot. Two things. One is, the idea of being able to find the models, be able to find the cancer in radiology may not be the right area. What he was saying was that radiology and pathology are facing major work shortages. So how do we use our expert radiologists? And maybe the idea is that the AI finds the normals and cuts out 70% of the workload for the radiologists and the pathologists, and that they focus on just that 30% where there's some ambiguity or there actually is some potential cancer cells there. So just cutting out the noise, maybe that in of itself is a victory.
I would agree with that. And there was that one slide that I showed that all the 9 true positives were caught by the model and 3 false positives were produced by the model. But that still is a 12 out of 55 workload where the AI system was able to eliminate 2/3 of the possible candidates. And so that's how I think we can bring these smart tools alongside to improve the efficiency of the clinicians, of the pathologists and radiologists. I'm also doing a model for lung cancer that's going to Malawi because there's only 4 pathologists for the whole country of Malawi. And so when somebody comes, we have a model that will detect the presence of lung or adenocarcinoma. So it's going to go over there to help the whole goal of being able to be a first stage, pre-sort the patients so that we can treat the most needed most quickly.
And then one of the difficulties with the pathology is not just the storage space, but also it's actually, even though it's flat and it's a slide, it still is a component of 3-dimensional because you go up, and when you're doing it on a regular microscope, you're kind of going up and down. So it's another layer that makes it a little more complicated than just strict imaging.
Yes.
But thank you very much, and thanks for your talk, and I feel smarter. So
thank you, Dr. Golden. Appreciate it. Any other questions? Let's see. I think on my first slide there, I'll put it back up. You know how to reach me. I'll through the department. Yes, sir. Okay.
How do you found representing lipids from protein interaction on carbohydrate interaction and all of that?
So yes, there is I sidestepped the real schema complexity. There's some very good research on ontologies for how to represent that. So there's some work that was done a couple years ago where they took just a lot of cases from cancer from the public TCGA cancer data set that NCI produces and analyzed them to figure out the types of the subtypes, and then very carefully created this graph. You have to, knowledge graphs I'm peripheral to that area. I'm not a specialist there, but I'm peripheral to it. And you take both the actual data and you take a schema, an ontology for, overall relationships, or types, and you say, lipid interactions or geospatial interactions because they're close. You need that hierarchy of ontologies also to be merged in so that you have a useful knowledge graph that doesn't confuse between the, like you said, the lipids and the other proteins, whatever. Yeah, and there are some well-crafted knowledge graph tools now And there's some open source tools that are good at saying, bring your data and just follow our steps, and we'll show you how to merge in these known ontologies. There's a medical ontology, there's a biology ontology. There's a lot of good work being funded to help. And so they're kind of mature enough now. It takes some time to do it, but Colorado, UC Colorado, I'll think of the name in a second. They were the initial people that did it. And now there's a couple, oncogenetics. I can find the ones if you're interested. But the answer is you do have to have the correct ontology so that you just don't generate just a bunch of random edges that confuse models instead. That's a good question. Well, I've come up on 4 o'clock here. I'll stay around for anybody who's in the room if there's any other questions they have. Thank you for the opportunity to listen to this talk, and I hope that it was helpful to you all in person and remote. Thanks so much. Okay.